All right, we are on now lesson 3 out of the 4 total lessons that make up this chapter, so we're almost finished. I came up with a super clever title for this one. It's called Why You Actin' Like That, because we're going to talk about a thing called actin. Guys, I was really proud of this. No one told me this. I just thought of this, just like with my own brain. This is as good as it gets. This is about as creative as I can possibly be. So the point of today's lesson is for you to learn how a muscle contracts. What's actually happening in your skeletal muscle to make it contract. So, on the first day, or even before we really started this unit, I gave you a piece of paper that had pictures on it. And the second picture is this one that's on the screen here. So you might want to take that picture out. Yours is not labeled, so you might want to add some things to that picture. So the title that I would give to the picture would be the word sarcomere, because that is what we are going to discuss. And so a definition of what a sarcomere is that it is the contracting unit in a myofibril. So to situate yourself where this actually is, notice that this top label here is a muscle fiber or a muscle cell. Remember, a muscle fiber is a muscle cell. In your first diagram, we have higher levels than that pictured, right? So many muscle fibers make up a muscle fiber bundle. Many muscle fiber bundles make up a muscle on the whole. So we're looking at one muscle fiber, one muscle cell. Each of them is made up of many myofibrils. And then inside the myofibril, there are these little protein filaments. So a sarcomere is the contracting unit. For you to identify a sarcomere, you need to find these zigzaggy lines here, which are called the Z line or the Z band. Kind of depends who's talking here. The Z something. Notice that they're zigzaggy. They kind of look like the letter Z. Makes sense that we gave it that name. So when you're looking at a myofibril and you're really zoomed in, you will see not just one or two, you will see many, many, many Z lines. Like they keep going and going and going. But in between two Z lines is a sarcomere. So between the Z lines, that is one sarcomere. There are many, 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 many sarcomeres in one myofibril. There are many myofibrils in a muscle cell. There's many muscle cells in a bundle. You get the picture. There are lots and lots and lots of them. Now this sarcomere, this is the thing that's going to actually accomplish muscle contraction, this tiny little unit. And so first we need to understand what it is made out of. So on this picture up here, you can kind of see that there's little lines, and some of them are thick and some of them are thinner. So I put this extra picture down here at the bottom to illustrate much more clearly, I think, what it looks like inside of the sarcomere. So notice here, I have some Z lines. It's the zigzaggy line. So that picture at the bottom is actually two sarcomeres. Inside the sarcomere, then, you have two different filaments. You have the thin filament, which is called actin. Actin is a protein filament. Filament means like a string or like a long piece of something. two key things here. Actin is thin. And I'm highlighting that because the other one we'll see is thick. The other thing, it's attached to the Z band. So on the picture at the bottom, you can see that the Z band and the blue lines are all one thing. So the actin is physically attached to the Z band. So these little things down here that look sort of like a tuning fork, I think, isn't that what a tuning fork looks like? Do people even use those anymore? Is that a thing in band? You like ding it and you get the tune? I don't know. A tuning fork? It's different, but it's the same idea. Anyways, that little tuning fork shape, that's the Z line, and then actin attached to it. So it's sort of a continuous thing. There's not a distinction there. Now myosin is the other one. Myosin is the thick filament. So it's visibly wider, thicker than the actin. It's found suspended between actin filaments, and it is not attached to the Z band. So we have a sarcomere. It's a section of the myofibril, and it's made up mainly of two types of protein filaments. Actin, which is thin, and myosin, which is thick. Now the picture that's in the middle will make a bit more sense once I show you what happens when the muscle moves. But the idea is that when a muscle is at rest, these dark spaces in the middle here, that's where the myosin is. And then there's some kind of lighter colored spaces here and here and here. Those lighter colored spaces are where there is actin only. There's no myosin in between them. And then as the muscle contracts, we'll see that those spaces sort of disappear. So we have the vocabulary. We have what a sarcomere is, that actin is a thin one attached to the Z band, and that myosin is a thick one not attached to the Z band. The next thing I'm going to show you is what happens when your muscle contracts. Now, there are pictures like the one that's going to be on the screen in your textbook, but I think it would be worth your while here to sketch out just a little bit of what's happening so that you can be sure you know what goes on in each step. Yes, Connor. Yep. Oh, it's an I. I'm not going to ask you to know all of that terminology. Like there's quite a few little, the I band and the M band and the H band and the zone and all this other stuff. The one of those that I want you to know is the Z band, because that's the ends of the sarcomere. Does anyone else have questions here? All right. So we're going to talk about the sliding filament theory. This is the mechanism for skeletal muscle contraction. Now the picture on the right is the detailed view. What I'm going to do is I'm actually going to draw it. So if I can draw this, you can draw it too. I'm going to draw what it looks like when the muscle, when we're sort of zoomed out a little bit on the sarcomere. So I'm going to start with two Z bands. They're very high tech. I know that Z bands are attached to actin. And I'm going to make sure that on this first picture, there's a space in the middle. The two sides of actin are not touching each other. So this is my actin, and that would be the Z band. Now the sliding filament theory says that actin filaments slide over the myosin. The myosin is here in the middle. It's thicker, so I'm making it look like a baguette, I guess. And like the picture on the first page suggested, there are little things sticking out of the myosin, which I'll talk about next, but I don't want to ignore the fact that they're there. So what I want you to notice in this first picture is that before the actin filaments slide over myosin, there is a bunch of space. See here, there's space. Here, there's space. And then in between all the actins in the middle, there is space. When your muscle contracts, what's actually happening is it gets shorter. And for your muscle to get shorter, without the pieces that make up your muscle breaking, we have to move them across each other and eliminate the space that's there. That's what's going to make your muscle shorter. So the sliding filament theory says actin filaments slide over myosin. That means actin moves and myosin stays essentially still. So what would my muscle, my little sarcomere, look like after muscle contraction had happened? Well, we'll go over here and give ourselves a little picture. First thing you should notice, it's not as wide. It's narrower than it was before. Second thing you should notice is now my actins are all touching each other in the middle. And my myosin takes up essentially the entire space that's available. So before my muscle contracted, I had all those black X's. I had space. Once my muscle contracts, I eliminate all of the space. The two sides of actin come closer to each other. That is the sliding filament theory. Actin filaments slide over myosin, and when they do that, all of that empty space is eliminated, and the muscle cell, and thus the muscle, gets shorter. Now, how does that actually happen? It's not like things are just sitting around sliding haphazardly for fun. There are specific mechanisms that cause the actin to move. So in the picture on the right, and there's a picture very similar to this on page 337 of your textbook if you'd like to look at it. But on the picture on the right, I'm going to illustrate to you what is going on at a really zoomed in level. So we are going to take like this little section here, and we are going to zoom in on it so that we can see what is going on. So I said myosin looks like it has things sticking off of it, and we're going to focus on those little things now. I'm going to start right here. On the top is actin. Actin is the little blue circles. On the bottom is myosin. Myosin is thicker, and it has the little golf club thing sticking up. 337. So here are some things I want you to notice on here. This little structure that looks like a golf club or a hockey stick is called a myosin head. Now, there's not just one of them. There's many, 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 many of these running the entire length of the myosin on the top and on the bottom. We're just focusing on one. The myosin attaches to actin. So for a muscle to contract, this has to happen. Myosin heads have to attach to actin. Now, what the myosin head is doing is very similar to what an oar does when you row a boat. So if you've ever been in a boat, a rowboat, or you've seen people rowing boats, when you put your oar in the water and you push on it, your boat essentially moves the other way from where you're pushing. So this myosin head is kind of like an oar. It's sticking to the actin and it's going to pull the actin back with it. So once it attaches, see how the myosin head bends. That little bend is called the working stroke, but the idea is that you stroke like you would if you were rowing a boat, so the little head bends back. And if you take a look, there's an arrow here indicating that the actin filament is going to move to the left on the picture. Now I want you to notice that ADP and PI were attached here. That's not just because, it's because at the next point down here, ATP has to come into play. So ATP is required for muscle contraction, but it's actually required for the thing that you would not expect. This ATP is required so that the myosin head releases the actin and then resets itself to its original position. So ATP is not realistically making your muscle contract. Moreover, it's letting it go. It's letting those two pieces of filament let go of each other. So perhaps you've heard of rigor mortis. Does anybody know what that is? It's when a person is... Oh, you want to say? Sure.
So when ATP is added
to the myosin head, yep.
Let's go.
Yep.
Let's go
Of the actin.
Oh, okay. So it attaches and then ATP is used to release it.
Yeah, to release it and to set it back up in the right position so that it could start again. So the ATP is necessary so that the myosin head will release the actin and the myosin head goes back to its original position so it can bind again. Yes? The ADP and PI are sort of leaving. They're going out so that they could, we would hope, become ATP once again with ATP synthase. So I'll go back to my point about rigor mortis. In rigor mortis, it's when a person is dead and their body feels really stiff. And if you think about the fact that ATP is necessary for muscles to let go of each other, if you're dead, you're probably not making ATP anymore. So ATP is not coming in. Your myosin heads could not detach from the actin filaments, causing the muscle to stay in this sort of permanent state of contraction. Does anyone else have questions that they want to ask here right now? Okay. So we're going to do two things. The first thing that I'd like to look at is just a summary of what calcium is doing. You saw it in the video. I made you act it out. But very specifically, here is what calcium does. The actin filament are these little round blue things all strung together. And covering the actin filament are these long strands of tropomyosin. Now when we acted this out, instead of having tropomyosin, I had all the actin turned the wrong way, so they couldn't see the myosin. We also have these little binding spots called troponin. They have a place for calcium to bind to. So when the calcium people came in and turned the actin the right way, we were simulating what calcium is doing here. When you add calcium, and calcium is this little white circle here, It binds to troponin, and it causes everything to sort of shift. What it does is it reveals the myosin binding site that tropomyosin was covering up. So tropomyosin would normally be covering those little yellow circles. That's where myosin wants to bind to actin. So calcium reveals the myosin binding site. That is its function. Now, if I ever asked you what is the role of calcium on a test, you could draw a picture to show me, or you could say it with words. Either way is totally acceptable, as long as I get the idea that calcium coming in turns things on the actin molecule the correct way so that actin and myosin can bind to each other. That is the main idea. Now, does anyone want to ask any questions about calcium's role, or maybe about any part of the sliding filament theory from the animation, from the notes, from our little acting it out? So then the last thing we'll talk about in notes today is how your muscle is acquiring all of this energy that it needs so that muscle contraction can continuously happen. So the last thing that we're going to take a look at is sources of energy. Now there's a picture on the right and words on the left, and the picture and the words are saying the same thing. We need to talk about where the energy is coming from. And energy is always talking about ATP. So in my picture, you'll see that every other way of getting energy eventually leads to ATP. You have to have ATP for muscle contraction, but there are a few different ways that you could get the ATP that you need. The first way is obviously ATP that is already made. So there was always some ATP being produced in your cell. There's cell respiration happening all the time. But there's not a lot of it usually just sitting around waiting for something to happen. But your cell would have some already produced, readily available ATP. Now notice I said this is used up very quickly. That's because there's not very much of it. So your muscle needs to have some backup mechanisms for how it's going to produce more ATP. So a second way that I could get ATP is from a substance called creatine phosphate. Now creatine phosphate very conveniently has a phosphate group. It will donate a phosphate to ADP, essentially producing ATP. So it's a quick way to get a third phosphate and get a molecule of ATP. On my picture, it's over here. On the top, it's showing creatine phosphate. Then it shows creatine plus phosphate, like the phosphate group has come off. And that's how I'm getting ATP. Your body makes creatine phosphate when it's at rest. So when your muscles aren't contracting, your body's making this so it has some of it stored up for future use. If you are at all familiar with supplements and like muscle supplements and things like that, you might see the word creatine a lot because this would be a way that you could help your muscles build more muscle tissue by giving them the energy that they need to contract. Now the next way is aerobic respiration. So when your body has depleted all the ATP that it has, and it's used up any creatine phosphate to quickly make some more ATP, now we've got to rely on cellular respiration to continue contributing. So on my picture, it's over here. The idea in aerobic respiration is that we start with glucose or some other type of fuel, but glucose would be the main one. Cellular respiration would happen. I would get a phosphate that I can attach to ATP through the entire process of cell respiration, which we've already discussed. I'm not going to go through that again. I would definitely end up making CO2 and water, but the result would be that I get ATP. Now, for aerobic respiration to occur, I need lots of myoglobin because that is where the oxygen is. Thanks. Now, the last thing that your body will do as a last resort, when ATP is gone, creatine phosphate is gone, and there is no more oxygen to be had, is it will rely on anaerobic respiration. So that's where we're making lactate or lactic acid. So we already discussed the mechanism of this. The idea would be, I'm in the middle here, only glycolysis will happen and then I'll ferment what I have. I'll produce lactic acid. I get some ATP, but not nearly as much. So that's the last resort. So these are places that your muscles can turn to when they require energy. So I'm just going to stop my video.